Spin-in-this-stiff microscopy can be a useful tool for studying the role of protein during cytoskeletal network formation. These techniques allowed three-dimensional imaging of fast cellular approaches that occur in the cell, and at the same time the precise localization of fluorescence molecules that are placed at the plasma membrane.
This method can provide insight into research areas like microbiology, cell biology, and all those branches where it's important to study the interaction between the plasma membrane and the cellular environment. This method can be extended to those live imaging experiments where it's important to localize the structures of the plasma membrane and where you also want to maintain a high resolution of the remaining cellular volume. The crucial things one has to consider are the choice of the proper cell line to set up the turf illumination and the other imaging parameters, and to find the focus of the transfected cells. The visual demonstration of this protocol will certainly help to avoid problems with the handling of the cells and during image acquisition.
Two days prior to the experiment, seed 300,000 HeLa or NIH3T3 cells in 2 milliliters of full growth medium into each well of a 6-well cell culture plate. The next day, prepare the transfection reagents. First, dilute 1 microgram of RFP Lifact and 1 microgram of YFP vinculin in a total of 200 microliters of reduced serum medium. Vortex the transfection reagent briefly. Then, add 4 microliters of the mixture to 200 microliters of DNA. Vortex the reagents again, and then incubate.